right, so welcome to my presentation for the doctoral colloquia. My name is Jana Černíková, my supervisor is Tomáš Vinaš, and the topic we are working on is from bioinformatics field. More precisely, we are interested in bacterial plasmids. So let me start with some biological introduction. In cells, either ours or bacterial, there is a molecule called DNA, which carries genetic information. And in bacterial cells, there are two types of such molecules. First is the chromosomal DNA, which carries the information that is critical for bacterial survival. And then there are plasmids, which are small circular molecules that exist independently of the chromosomal DNA. Why are the plasmids important is that they carry beneficial genes for the bacteria, for example, those for antibiotic resistance. And they can transfer between the bacteria. For example, here we can see that the bacteria can somehow connect, and the plasmid DNA is copied to a bacteria that did not contain plasmid before. We call this transfer horizontal gene transfer. And then there are new bacteria that have the plasmid that also carries the antibiotic resistant genes, which is an issue. Also, when the cells replicate, they replicate their chromosomal DNA, but also the plasmids can get to the new cells. Another thing that may happen is called plasmid integration, and it causes the thing that the sequences that were originally found in plasmids will get to the chromosomal DNA. And this way they can be somehow evolutionarily conserved, which causes some issues that we will see later. Now, how do we get the information from such bacteria if we want to analyze them computationally? This process is called sequencing. So we have some DNA molecule in the bacteria or another organism, and then using some sequencing device, there are many platforms for sequencing, we can get this information to the computer. We are working specifically with nanopore data. Why is it called nanopore? Is that the molecule traverses through a small pore, which causes changes in electric current, which are then measured and translated into sequence of A, C, G, and T's in process we call base calling. And the result of the sequencing experiments are the sequencing rates. Here is a toy example. We have some header with some metadata information, and then there is sequence itself. And we are working with such data. So what do we do with such data is, for example, genome assembly. And even though the genetic information that is in bacteria is there in nicely tight form, when we sequence it, what we get is more like a shredded pile of paper. And what we would like to accomplish is to get the individual pages from it. However, it is not like this all the time. Commonly, we get something more like pieces of those pages, often split in a very confusing way, and we still somehow want this page, and we call this fragmented parts contigs. And further, when we look back at our bacteria, we have two types of pages, one for chromosomal reads and one from plasmid reads. And therefore, we also have two types of shredded paper piles. So we have some reads that are chromosomal and some reads that are plasmidic. But unfortunately, in our data, they are mixed together. And what we can do is ignore the fact that they are mixed together and somehow assemble the contigs from all the data we have. And Then another task we have, and this is a very different and challenging task, is to label the contigs based on whether they originate from plasmids or whether they originate from chromosomal DNA. So there are many approaches how people do this. For example, there are machine learning methods which people commonly use. Then there are graph-based approaches which operate on graphs constructed from the reads and their overlaps. Then there are hybrid or heuristic approaches which commonly combine machine learning and graph-based methods, and also another heuristics. And there are also alignment-based approaches which somehow use the databases of known plasmids. And they align the sequences to them. And this way they can get some information about the reads. Our main focus is on the machine learning methods because they generally outperform the others. And the machine learning methods in this case often use regression, decision trees, support vector machines, neural networks, or deep learning. And they are implemented by many different tools. But all of the tools we have found operate on assembled contigs, which are very long sequences. And there is only a small number of them for a sequencing experiment. And what we would like to do is to classify the individual reads. This is a harder task because there is a lot of the reads, and they are generally shorter. So we want to somehow split this pile into the two piles as we seen before. And another way how we categorize the tools is whether they are species agnostic or species specific. The species agnostic tools operate on multiple species, where they can get data from many species, and they will output whether the contig is plasmidic or chromosomal. Then there are species-specific classifiers which operate on data they were trained on, or on the species for which they had the data. In general, it is more difficult to design the species-agnostic classifier. That's why we have started with the simple option. And the natural question that arises here is what will be our labels and features, and how do we obtain them? So we have our read, and we would like to get some meaningful information from it. And for the gram truth labeling, we can use the before mentioned classification tools. So we take our data and assemble the contigs. Then we can label the contigs based on the classifiers we can use. And then we somehow want to split this pile using this information. So this is the result we would like to get. One method how we can do this is called sequence alignment. So we have some sequence which is known to be plasmidic. Then we have some sequence which is known to be chromosomal. And what we can do is to look for some similarities between the plasmidic contig and the reads that are similar to it. And this is done using dynamic programming according to some scoring scheme, as we can see here. So we have two sequences, and we have some scores for matches, which is positive, some score for mismatches, which is negative, and we want to include some gaps into the sequence so that the score is maximal. Also, the gaps can have some slightly negative score. And then we somehow obtain the alignments. And maybe we could say that, okay, if the read aligns to the plasmid contig, let's label it as plasmid. If it aligns to the chromosomal contig, let's label it as chromosome. But it's not that easy, because there is an issue, and the issue is that a lot of the reads align to both plasmid and chromosomal contigs, and also the alignments are local, meaning partial. And therefore, we have designed something we call plasmidness and chromosomeness score. And how they work is that we classify read as plasmid if the plasmidness score is more than 80 and chromosomeness score is less than 20. And similarly, we do this for chromosomes. If the read does not meet any of these criteria, then it is discarded from our data set. So how do we obtain the scores? We have this contig, and we have a read, and this read aligns to 3 places. The first place is on the plasmid contig, and this orange part is similar to the orange part in the read, and this green part is similar to the green part of the same read. And then we take the union of the intervals that are covered by the alignments, and using the length of the union interval, we compute the percentage of the read that is covered by the alignments, and that is our plasmidness score for. So, for example, here it might be 80%. And the same read may also align to the chromosomal contig. Let's say this purple part is similar, and then we would say that okay, this is 15%, so the chromosomeness score will be 15%. Then, based on our rules, we have plasmidness score which is over 80, and chromosomeness score which is less than 20, and therefore the resulting label will be plasmid. Now let's look at our features. The first feature we have is the relative average coverage by the other reads. And how do we compute it? Is that we take a read. For this read, we take all other reads from the sample, which is sequencing sample. And we align the reads to this particular read. And for each position, we compute how many reads aligns to that position. And then we take the average of all the positions in the read. So for this example, we would get average coverage approximately 3. But since the coverages for samples are different, and we have multiple sequencing samples from multiple sequencing experiments, we further divide this value by the average of all coverages for reads in the sample. So we somehow normalize this value. And then the features we use are the GC content, which is basically the percentage of bases that are G or C. Then we use read length, and then we use average read quality, which is the information we get from the base color, which reads the signal from the sequencing process. And the base color assigns each of these letters it reads a quality value, which basically says how sure is the base color about the particular base. And then we take average of these scores. Another feature, or better said, set of features, is the K-mer profile. The big drawback of this set of features is that they are highly species specific. And so the K-mer is basically a string of length k. We choose the number 5 because many papers state that 5 is the best number among reasonable options for some reason. And for k equals 5, we get 1024 possible 5-mers over our alphabet of 4 letters, which yields 1024 possible features, each being a frequency of 1 5-mer. Here is an example of the K-mer profile, but not for a single read, but for the whole training set. As we can see in this picture, here we have the training set. Here we have the testing set read. And each square in this plot corresponds to the frequency of 1 5-mer. And if we look at these plots, they are somehow similar. You can see the darker part here. It is all darker also here. For example, here too, here too. So they are somehow similar, which suggests that this might be some informational feature. Another thing we have found is that it highly depends how we divide our data. This is because we have multiple samples, or better said, isolates of E. coli bacteria in our samples. And each sample has different portion of chromosomal and plasmidic reads. And also the number of reads in the samples varies. Here, but what is common among them is that there is only a small number of plasmid reads in general, so we have a highly imbalanced data set. Here is an example on our real data. The red bars are the counts of chromosomal reads. The blue bars are counts of plasmid reads. And as we can see, that the number of plasmid reads is generally lower than the chromosomal. Those are the first 10 of our training samples. So what we have done so far, we have trained multiple simple neural network models on two sets of features, on two sets of isolates, using our features and labels. Then we have evaluated precision recall and F1 score for them. And we have found that we can get good results if the isolates are mixed. So if we randomly select 80% of the data set for training, and 20% for testing, then we can get good results. But if we divide the data set in a way that we have 9 training isolates and two isolates for testing, then the recall for plasmid was very low. Then we tried to increase the data set size, which gave us higher plasmid recall. And then we also experimented further with this larger data set. And also we experimented with the network architecture. So here are the results for some of our experiments. As we can see here, for the mixed isolates case, the recall and precision for plasmids is high. This is because in this case, the model is able to learn exact sequences and then classify them. But in the second case, where the isolates were not mixed, then the recall for plasmids has dropped significantly. And the reason for this is that there is a large difference between the training and testing data set. We've tested this hypothesis by comparing the data we have in the training and testing set, and found out that there is only 40% overlap between the testing and training contexts. Then we have tried to enlarge the data set. This way we got a higher recall score, but still for the same reason as in the previous line, the recall is still low. Another thing, we tried to alter the architecture a little. So we have added a narrow layer in the middle of the architecture, trying to avoid learning of exact sequences, but the recall here increased only a little bit. And another experiment we did is that we decided to classify the long reads only. And this way we were able to get 80% recall, which is the best result we get so far. In bioinformatic context, setting such threshold isn't that bad, because what we care the most are the long reads. In future, we would like to design new features that may help distinguishing between chromosomal and plasmidic reads. For example, we can use homologies with non-plasmidic or chromosomal sequences. Then we would like to train different models, for example, to avoid the memorizing of exact sequences. So we can maybe change the width of layers or model depth. And we also want to use additional information from read overlaps. which can be done using graph neural networks. Another direction is to focus on less species-specific features, which we also plan to do in future. And if you look at the homology features, which is something I started working on, we can see that on this plot, there is on x-axis something we call chromosome score, which we get from alignments to some known sequences. And then on y-axis, we have plasmid score, similarly as for the chromosome. And we can see that this feature can separate the data pretty well. So we have the red are chromosomes and the blue are plasmids. So this direction looks promising. So to summarize, we have designed an approach to label the sequencing reads based on the information we have. We have proposed some features that can be used for read classification. We have trained multiple neural network models for plasmid read classification and evaluated the commonly used metrics. Then we have investigated the causes of why the recall flow plasmid was low and proposed some future directions. Thank you for your attention, and questions as to the scenario.